Have you ever wondered what makes something a human embryo, especially when it comes to scientific exploration? It is a question that sits at the intersection of biology, ethics, and societal values. For research purposes, the definition of an early embryo typically begins with fertilization, the creation of a zygote, and extends through the pre-implantation stages, primarily up to the blastocyst stage. This often includes a widely accepted ethical benchmark of up to 14 days of development. The why behind these definitions is rooted in both biological development and profound ethical considerations. A zygote, formed from the fusion of an egg and sperm, is the earliest stage, containing a full set of human chromosomes. It then rapidly divides, becoming a morula, a solid ball of cells, and then a blastocyst, a hollow ball structure that forms about 5 to 6 days after fertilization. These early stages are crucial because they contain pluripotent stem cells, which have the remarkable ability to develop into almost any cell type in the body, but not an entire organism on their own. The 14-day rule, or the primitive streak rule, is a key ethical standard recognized internationally. It marks the point before the primitive streak, an early precursor to the nervous system, begins to form. Prior to this, the embryo has not yet committed to forming a single individual, retaining the potential for twinning. This benchmark attempts to balance the potential for scientific discovery with the emerging moral status of the developing entity, acknowledging that beyond this stage, individuality becomes more established, and ethical concerns deepen significantly. The definitions are critical to guide research ethically and ensure clear boundaries. So, how do researchers and ethical oversight bodies apply these definitions in practice? This is where institutional review boards, or IRBs, and national guidelines come into play. When a scientist proposes research involving human embryos, they must submit their plan to an IRB or a similar ethics committee. These committees rigorously review the proposal, ensuring that it adheres to established definitions and ethical limits, such as the 14-day rule. Researchers must demonstrate that their work uses the earliest necessary stages of embryonic development to achieve scientific goals, minimizing ethical concerns. This structured oversight helps ensure accountability and adherence to agreed-upon ethical boundaries, making sure that scientific advancement proceeds responsibly. An interesting concept tied to this is the distinction between totipotency and pluripotency. A zygote is totipotent, meaning it has the total potential to form an entire organism. As the embryo develops into a blastocyst, its inner cell mass contains pluripotent stem cells, which can form all cell types of the body but cannot, by themselves, develop into a complete organism. This shift in developmental potential is a key biological factor that informs ethical discussions and research guidelines. Ultimately, defining early embryos for research is not just a biological exercise. It is a dynamic process shaped by scientific understanding, ethical deliberation, and societal consensus. These careful definitions provide a framework that allows for groundbreaking research into human development and disease, while upholding important ethical considerations. It is a continuous dialogue, ensuring that our pursuit of knowledge is always balanced with our moral responsibilities toward human life at its earliest stages.